Digestion of carbohydrates, such as starch found in the crust of the piece of pizza, begins when the carbohydrate is taken into the mouth and begins. Chewing acts to physically break down the food and mix the carbohydrate starch with alpha amylase found in the saliva. It should be noted that the saliva amylase is not as active as the pancreatic amylase found in the small intestine. After chewing, the food is swallowed.
The food travels down the esophagus through a process called peristalsis, which is the contraction of smooth muscle, and ends up in the stomach. Not much in regards to carbohydrate breakdown happens in the stomach, but the food is then turned into chyme. The chyme passes through the pyloric sphincter and ends up in the duodenum, the first part of the small intestine, for carbohydrate breakdown.
Good.
When chyme reaches the duodenum, it is mixed with bile. Bile is produced in the liver and stored in the gallbladder. The role of bile is to emulsify fats and also buffer the highly acidic chyme before it moves further along the digestive tract.
Okay, once the chyme enters the small intestine, it's subjected to digestion by a host of enzymes, including alpha amylase. The role of alpha amylase, which is produced in the pancreas, is to break down the alpha 1,4 bonds between the glucose moieties and starch. Starch is a homopolymer made of glucose. Digestion by alpha amylase produces limit-dextrin and maltose. Limit-dextrin contains an alpha 1,6 bond, which cannot be broken by alpha amylase. It is instead broken down by alpha dextrinase into maltose. Maltose is then broken down into glucose molecules. Maltose is digested by alpha glucosidase to produce glucose molecules. The enzymes mentioned are located in the brush border of the small intestine. It should be noted that the brush border also contains sucrase and lactase, which break down sucrose and lactose respectively.
The brush border is made up of small finger-like projections called microvilli that increase the surface area of the small intestine to aid in the absorption of monosaccharides.
Once starch is broken down into monosaccharides, the monosaccharides are absorbed by the intestinal cells and transferred to the bloodstream. Glucose and galactose move into the intestinal cell out of the intestinal lumen via the sodium-glucose link transporter. As sodium diffuses down its concentration gradient, it brings with it glucose and galactosidase. Fructose, another monosaccharide, diffuses into the cell via GLUT5. Once in the intestinal cell, the monosaccharides are transferred to the bloodstream via GLUT2 through diffusion. Once in the bloodstream, the monosaccharides can travel to the tissues in need of carbohydrates as an energy source.
Now we are in the cytoplasm of the cell. This is stage 1 of glycolysis. The transfer of the phosphorus group from ATP to the hydroxyl group on carbon 6 of glucose is catalyzed by hexokinase. This reaction is a phosphorylation. The isomerization of glucose 6-phosphate to fructose 6-phosphate is a conversion of an aldose into a ketose. The reaction is catalyzed by phosphoglucose isomerase. Fructose 6-phosphate is now a pentose. Fructose 6-phosphate is phosphorylated by ATP to fructose 1,6-bisphosphate by phosphofructokinase, an allosteric enzyme that is the key regulator for glycolysis. Fructose 1,6-biphosphate is splitted into two triose phosphates, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, by the enzyme aldolase. Both compounds are isomers that can be readily interconverted in an isomerization reaction catalyzed by triose phosphate isomerase. In stage 2, a redox reaction converts glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. This reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate is oxidized and NAD+ is reduced to NADH. Phosphoglycerate kinase catalyzes the transfer of the phosphorus group from the acyl phosphate of 1,3-bisphosphoglycerate to ADP. ATP and 3-phosphoglycerate are the products. 3-phosphoglycerate is converted into 2-phosphoglycerate by phosphoglycerate mutase, which shifts the position of the phosphoryl groups from the third carbon to the second carbon. The dehydration of 2-phosphoglycerate catalyzes catalyzed by enolase, introduces a double bond, creating an enophosphate. Enolase catalyzes the formation of the enophosphate, phosphoenopyruvate. When the phosphor group has been donated to ATP, the unstable eno is able to undergo a conversion into the more stable ketone, named pyruvate. This step is not reversible and catalyzed by pyruvate kinase. At the end, 2 pyruvates, 2 ATP, 2 NADH, 2 H+, and 2 HO2 molecules are produced.
Fermentation releases energy from food molecules by producing ATP. Glucose is broken down through the process of glycolysis, which then breaks down to pyruvate, which nets 2 ATP. Then pyruvate is broken down to 2-acetaldehyde, and in this process it releases 2 CO2. Then the energy from the 2 NADH is used to break down acetaldehyde to ethanol, and this process is called alcohol fermentation. Glucose again is broken down through the process of glycolysis, which then it breaks down to pyruvate, where it nets 2 ATP. Then pyruvate is broken down to lactate, and it does this by taking the energy that was stored into an NADH, and this process is called lactic acid fermentation.